Assalamualaikum and good day everyone. So this is the fourth part of the adaptive lecture for adaptive immune response. So this part 4 we will talk on antibodies. Antibodies is the immune system smart missiles. It is a specific protein produced by B cells in response to adaptive immune system response to an antigen. When the body face a pathogen, a particular pathogen for the first time, the B cells will produce specific antibodies that specifically will recognize that pathogen by looking on the associated antigen. Specific antibodies will bind to epitopes on the specific antigens. Epitopes is like the structure on the main antigen. So antibody works by forming a lock and key mechanism where they will be able to combine specifically with the epitope on that antigen, which then eventually forms antigen antibody complex. Now we look on the antibody basic structure. Antibody consists of 4 protein chains held together by a disulfide bonds. The bridging between the structures. The 2 largest chains are identical to each other and are called the heavy chains. So the light and the purple and the light purple area is the heavy chain. And 2 smaller chains are also identical to each other and are called the light chain, the green part. They are joined together, the heavy and the light chain forms a basic Y structure. This is a typical structure of an antibody. So now we look on the parts that giving the function of the antibody. So we have the Fab region, which is the fragment antigen binding site, the part that bind to the antigen. Here they are consist of the variable region, the variable region changes to various structures depending on the differences on the antigens. So the variable regions are designed specifically to fit on the shape of the antigens. Give the antibody its specificity for binding that antigen, and they also consist of a constant region. Constant region determines the mechanism or the way how the immune system will destroy the bind antigen. Includes the trunk here and lower portion of the each arm of the Y. So the dark green and also the darker part of the purple includes in the constant region. Antibodies are divided into different classes. Here we have 5 classes of antibody: IgG, IgM, IgA, IgD, and IgE. 5 classes. This based on their unique constant region structures. So how we classify the antibodies into 5 different classes is based on the shape of their constant region of the heavy chain and also their immune function. Second part that giving the function on the antibody is the FC region, the fragment for crystallizable. This is the part that active the immune response. This is the site of complement binding binding the complement factor, and also binding to phagocytic cells, FC receptors during antibody mediated opsonization. So how antibody works? Antibody mediated responses are especially effective against first, extracellular pathogens, and second, antigens that dissolve in body fluids such as toxins and also viruses. Antibodies provide immunity in three ways. So how they contribute to the immune system? First, by preventing entry, so preventing pathogens from entering or damaging cells. Second, by stimulating removal or discard the pathogens by macrophages and other cells. And third, by triggering destruction of the pathogen. They performs that first by neutralization. Neutralization is the binding of antibodies on epitopes on the surface of pathogens or toxins, preventing their attachment to cells. Some works as antitoxins where they block toxins, example that those that causing diphtheria. For example, here the antibody is blocking the diphtheria toxins released by Bordetella pertussis bacteria, and also tetanus. Second, by opsonization. It is the binding of antibodies on epitopes on the surface of pathogens. This is to help the phagocytosis process. These antibodies will attach some antibodies attach to bacterial flagella, making them less active and easier for phagocytes to engulf. As we can observe here, phagocytic cells such as macrophages, dendritic cells, and neutrophils have the FC receptors on their surfaces that will recognize the FC region on the antibody. The macrophages will attach its FC receptors to the FC region of the antibodies. This helps the phagocytes to attach and engulf the pathogens bounded by the antibodies. Third, we have agglutinating and precipitating antigen, where it causes agglutination or clumping together of bacteria, making them less likely to spread by limiting its movement. Involve the cross-linking. Cross-linking means the attachment of between two or more pathogens by antibodies to create large aggregates, where we can observe here in the picture the pentameric structure of IgM. So IgM, majority of IgM consists of five antibodies attached together to form a pentamers, where they have a 10 FAB here, 5 times 2, making it the most efficient antibody for agglutination. So they will have 10 site for attachment. Other antibodies like IgG monomers that exist in a single form, has two Fab antigen binding sites, but they still can bind to two separate pathogen cells, clumping them together. So when multiple IgG antibodies are involved, still can produce large aggregates of antigens. Fourth, by activating the complement proteins. The complement system is an important component of the innate defenses. Among the benefits include promoting the inflammatory response, recruiting phagocytes to site of infection, enhancing phagocytosis by opsonization, where complement also can help phagocytosis by the opsonization process, just like antibodies, and also by killing of gram-negative bacteria pathogens by making a hole on its cell surfaces by forming the membrane attacks complex. And fifth, the antibody-dependent cell-mediated cytotoxicity, or ADCC, where it enhances killing of pathogens that are too big to be engulfed by the phagocytes or other cells. This process is best characterized by the natural killer cells, but can also involve the other macrophages. ADCC occurs when the Fab region of an IgG antibody bind to a large pathogen. The Fab region here, the fragments of antigen binding attached to the epitopes on the pathogen. The Fc receptors on the natural killer cells then bind to the Fc region of the antibody, so like a lock and key mechanism. So this will making it easier for the natural killer cells to attach to the or come near to the pathogen, bringing them into close proximity with the target pathogen, which will able them to secrete powerful cytotoxin that will kill the pathogen because of the close gap between them. This will enable the NK cells to release the cytotoxins in order to kill the pathogens. The 5 classes of antibodies: IgG, IgM, IgA, IgD, and also IgE. First, we look on IgG. The majority of antibodies is in the form of IgG, around 80% percentage in plasma. They can be found all over the body. The function include neutralization, agglutination, complement activation, opsonization, and also ADCC. The special abilities of IgG: they are the smallest, so they can penetrate efficiently into tissue spaces, and is the only antibody that can cross the placenta. Means it can the pregnant mother able to protect the developing babies, fetus, by sharing its IgG with the fetus. Second, IgM. IgM can be found in a pentamer form, where 5 monomers attach to become one molecule of pentamer. So IgM function in neutralization, agglutination, especially in agglutination, because the pentamer is very efficient in performing agglutinates, and also complement activation. They also exist in monomer form. The monomer forms serve as the B cell receptor on the B cell surface. The IgM is the first antibody produced and secreted by B cells during the primary and also secondary immune responses, because they are the biggest pentamer with high avidity. They have 10 combined binding sites compared to the other type of antibodies. Third, the IgA, where can be found in the body secretions. They exist a majority in dimers, where two monomers attach to become a dimers. Function in neutralization and trapping of pathogens in the mucus. IgA protect body surfaces that exposed to outside foreign substances, such as the mucosal surfaces. IgA from breast milk, in the mother's breast milk, can be transferred to the baby, and this can protect the baby against infections in the early months after child delivery. Fourth, we have IgD. IgD majority can be found on the B cell surfaces. It is a membrane-bound monomer found on the surface of B cells, where it serves as an antigen receptor. IgD acts as a B lymphocyte activation signal, and they also found to play some role in allergic reaction. And the least common type of antibodies can be found in our bodies, the IgE, which is less than 0.05%. The majority of IgE is on the surface of attached to the surface of mast cells and basophils, and they can be found in the lung, skin, and mucous membranes. IgE, the main function is to activate basophils and mast cells in response against parasites and also allergens. They stimulate mast cells and basophils to release a very potent pro-inflammatory mediators, and they also one of the main culprit in stimulating the allergic reactions.